I'm here with Dr. Pablo Zamora, here to talk about metagenomics. Now, my first question for you is, what is metagenomics?
Okay, hi Rex. Metagenomics is a brand new technique that the idea is try to get sequencing to study microbial community most of the time without culture. The idea first is try to get a really nice picture about the uh, environmental diversity. Before to metagenomic analysis, we just pick up some of the microbes that live in environmental with culture. That means that the microbes that live in the culture, they can grow. But doesn't mean that the bacteria is really important in that environmental. With metagenomic data, it's like we take everything and we make for that analysis. The thing is, we take sequence for that microbes and we make a whole spectrum about the environmental. It's a really cool system. Yeah.
So this has really impacted the scientific community?
Yeah. Because now we can know much more about the real diversity. For example, in the human gut, normally we know that a really small portion of the microbial diversity we can study because we can culture it. But now we can study the whole community. That means that we are discovering new species, new bacteria, new function, and new metabolic pathways. And the metabolic pathways more or less are related with some physiological features like, diseases or some traits. That means that it's fantastic.
So with this, you can really help the world be a better place, huh?
Yeah, because if we have more understanding about the diversity, the microbes or their life, we know better how to treat the diseases, we know how to improve the agricultural stuff and these kind of things. Yeah, it's really huge impact too.
Since this is such a new community, metagenomics, how does this create problems and difficulties for your research?
How they create problems itself?
Yeah, because it's so new, you have to think of all these new techniques.
It's quite, it's really good your question because we have two problems. The first one, that the metagenomics is a really expensive tool in compared with normal tools that we just pick up some bacteria. For example, we grow in a media culture. In this case, we need to make a how-to-put sequencing. That means that we need to get higher amount of DNA to get a profile for the bacteria. And also, the idea then is not just pick up the DNA and analyze the DNA. We need to use the information to get some result. And that part between the sequencing and the analysis of the data is huge. That means that you need a lot of facilities and people like bioinformatic people that can work with the data and probably take some result out.
Okay. So, what does your typical day consist of working at the lab?
Yeah, working at the lab, we work in two different areas. Our work here in UC Davis is related with plant microbe interaction. Some of the day I'm in the middle of the jungle in Mexico studying plant microbe in corn. And it's quite exciting because we work with indigenous culture and then we bring the samples and here we make the metagenomics there in that building. That the name is the Genome Center from the UC Davis. Then we take the samples, we run the analysis, and I come back to the lab and with another collaborators, like people from different labs, we take the data and we start to make the analysis. That means that we extract the DNA, we see the profile of the DNA, and then we try to understand the data. Normally, I take it in my computer and I run software and I think and I read papers just to understand which information can be useful for our goal, that is plant microbe interactions.
Okay. So metagenomics is really important for your research. It's for microbial communities because recently you've discovered so much more about these communities that just haven't been possible before.
Yeah,
so,
exactly. Open a huge
new door.
Yeah, that is, everything inside the room is new.
And what kind of stuff do you get to get your hands on, that you get to work with to discover these new things? What kind of techniques do you use?
We use different techniques. For example, in the case of the metagenomic data, that is genomics is related with the DNA. You have also like the sister and the brother of the metagenomics is the metaproteomics. That means that at the same level you may have two-put sequencing, but not DNA. You work with proteins, and also you have the metatranscriptomic data. That means that you work with the RNA. That means that we take all the samples and we understand much better what is the diversity, what is the expression of the gene, and what is the function of that community. And everything is complemented with another technique, that is metabolomics. You know that the meta, that means that it's the how throughput, the high amount, like ridiculous amount of data at the same time. And we are making some really nice discovery because we know, for example, in our research that how some particular bacteria can feed. Maybe it's not interesting for most of the people, but for us it's really important because we are understanding how some microbes can use a plant exudate to feed some communities. How is this one is related for the beneficial part of the plant? That means that it's really nice to see a picture, but then you can get a movie if you use the metagenomic, metatranscriptomic, proteomic data, and also metabolomic. That means that you can understand how the cell functions.
Okay, and what got you into this field of science and research and metagenomics?
That question is really hard to understand for me. I think that the first thing is because I really want to know what's happening outside of in the environmental and how we can understand the life. This is the first thing. I started when I was really younger. And my first approach was started really the chemical part of the life, like the biochemistry. And after the biochemistry, I jumped to the how throughput sequencing. That means that try to understand how the cell can be alive, how the cell can be transmitted, how the cell communicate to each other, and then understand how they organize. Yeah, that was a really long process. It was not from one day and say I want to make metagenomics and that's it.
It was more complicated than that.
Yeah.
Well, thank you for your time. We learned a lot about metagenomics. So I hope that with what you've said, that more people can do a little bit of research about metagenomics.
Yeah, and everybody's welcome to visit our facilities here in UC Davis.
UC Davis. Well, thank you very much.
Thanks, Rex.